Manjula Bomma, currently affiliated with Alabama A&M University, where I serve as a part-time postdoctoral researcher and adjunct faculty member. I hold a PhD in microbiology and I have been involved in postdoctoral research since completing my doctoral studies. My research encompasses several critical areas in microbiology with a particular focus on the antibacterial properties of medicinal plants. Additionally, I have worked extensively on the synthesis of various nanoparticles and their innovative applications in molecular biology, such as gene transformation techniques. My work prioritizes environmentally friendly and biologically enhanced methods for nanoparticle synthesis, with demonstrated effectiveness in antibacterial studies. These efforts have led to multiple peer-reviewed publications that contribute to sustainable and impactful microbiological solutions. Recently, my research interest has expanded into the field of low temperature plasma, inspired by its eco-friendly nature and promising potential in microbiology. This cutting edge technology opens new avenues for developing non chemical, sustainable antimicrobial approaches for ensuring plant and food safety. I'm excited to explore its innovative applications further in the context of microbial control and biotechnology. Throughout my postdoctoral and doctoral journey, I have had the privilege of working under the mentorship of Dr. Okafor and Dr. Mentreddy, whose guidance has been instrumental in shaping my research direction and academic growth. My published research on bioproduced nanoparticles and medicinal herbs on human and plant pathogens. In 2019, we published enhanced silver nanoparticle synthesis by Escherichia coli transformed with Candida albicans metallothionein genes. In this study, we have used PUC19, where the transformed gene we introduced in PUC synthase gene. And here we have seen the heavy metal toxicity on the copper. We observed the more production of the metallothionein gene synthesized E. coli have the more cell growth rather than controlled one. And in 2020, we published expression of Rhizobium tropici phytokeratin synthase in Escherichia coli resulted in increased bacterial selenium nanoparticle synthesis. We produced here using the Rhizobium tropici like phytokeratin synthase we used to develop selenium nanoparticles, where we have done the same activity but more on the heavy metal toxicity by seeing like copper, nickel and zinc and cadmium, like heavy metals. In 2021, we published enhanced extracellular synthesis of gold nanoparticle by soluble extracts from Escherichia coli transformed with Rhizobium tropici phytokeratin synthase gene. By using the same phytokeratin synthase gene, we produced gold nanoparticles in this study. And here we have used intracellular and intracellular extractions of the gold nanoparticles, and we used two different spin productions like 3000 RPM and 17,000 RPM, two kinds of speeds here. And the last publication from my as a co-author, as a first author, comparison of phytochemical comparison, free radical scavenging activity and antimicrobial activity of selected herbs against two food-borne pathogenic bacteria. Here, I just selected two medicinal herbs like turmeric and mountain mint, where I did antioxidant activity like DPPH assay, where high antioxidant activity shown by the turmeric, and MIC MBs are more effectively showed by the turmeric. Today, my presentation is assessment of atmospheric pressure plasma for inactivating the seed-borne pathogen Xanthomonas campestris, Pathobar vesicatoria on bell pepper seeds. Outline of this study is introduction, material and methods, bacterial strain and culture conditions, cold plasma treatment of bacteria, bacterial growth rate measurement, data analysis, results, discussion and conclusion. The objective of the study is assess the effectiveness of atmospheric pressure plasma in suppressing seed-borne bacterial plant pathogens. Seeds are vital to global agriculture with nearly 90% of food crops originating from them. However, seed-borne pathogens like fungi, bacteria, and viruses pose a serious threat to crop yield and food security. Bacterial leaf spot caused by various Xanthomonas species is a major disease in warm, humid regions, especially affecting solanaceous crops like bell peppers. It spreads mainly through contaminated seeds and can result in up to 40% of crop loss, making its management difficult due to the pathogen's genetic diversity and adaptability. The images shows bacterial spot disease symptoms caused by Xanthomonas species and pepper and tomato plants, and the infected leaves are the necrotic spread on the leaves. And the map shows about the illustrates the distribution of the different Xanthomonas species causing bacterial leaf spot in solanaceous crops. Each colored symbol expresses a specific species, like different species all over the globe. These pathogens are found across the continents, including America, Europe, Africa, etc. And it is a widespread in nature and global threat of the BLS to crop production, especially in solanaceous crops like tomato and pepper. This figure D shows like main seed treatments applied to control seed, fungi, bacteria, and viruses. We have different kinds of treatment on the seeds. Those are the natural compounds we can use for the seed treatments before the cropping, before it is going into the field, and biocontrol agents and fungicides. And physical, when you talk about the physical treatment, cold UV radiation is surface limited and potentially damaging with overexposure. Heat treatments are effective but often stressful for the potentially reducing germination and vigor. Coming to the cold, plasma is non-destructive, chemical free, and offers broad spectrum antimicrobial activity without harming seed viability when properly optimized conditions. And atmospheric pressure plasma is a non-thermal, eco-friendly technology, and broadly we can use in the applications like agriculture, the food industry, and medicine. And in the decontaminating spices, liquids, and seeds, enhancing shelf-life, preserving food without compromising quality. In the medical field, it plays an important role in sterilizing instruments and masks and promoting wound healing and cosmetic treatments, removing dental plaque, and even targeting cancer cells. Its versatility makes it a valuable tool for safe, chemical-free microbial control and therapeutic applications. The image shows how the plasma treatment affects pathogens and plants, and in the direct treatment uses UV ions, electric fields, and ROS and RNS groups like reactive oxygen species and RNS for the reactive nitrogen species, while indirect treatment mainly involves ROS RNS groups, where it inactivates the pathogens and it will directly attack DNA damage and lipid peroxidation, so that it is called as a plasma vaccination. This image illustrates the four states of matter: solid, liquid, gas, and plasma. As energy is added, matter transitions from solid to liquid, liquid to gas, and gas to plasma. Plasma, the fourth state, forms when gas particles gain enough energy to ionize, creating a mix of electrons and ions. This high energy state is seen in lightning stars and plasma technologies. This image illustrates about the interaction between plasma and liquid interface chemistry and ROS groups, how they can reactive in the biological medium. So these species penetrate through the interface layer where they fuse into the bulk liquid or biological medium, triggering complex chemical reactions and producing RONS and ROS groups. And this mechanism is fundamental to plasma medicine, water treatment, and sterilization applications. The image demonstrates how plasma inactivates microorganisms like bacteria and enveloped RNA viruses. Plasma generates ROS RNS groups where they can act upon the microbial structures by damaging the envelope capsule, RNA or DNA, ultimately leading to bacterial inactivation. Coming to material and methods, bacterial strain and culture conditions. Aseptically transfer bacteria to solid nutrient agar. Incubation for the 24 hours and 28 degrees centigrade for the Xanthomonas campestris pathover vesicatoria. We have selected single colony and incubate in 5 ml nutrient broth and incubator for the 24 hours. For the following day, we have done the microdilution for 10 to the power of 6 CFU per ml and added 0.6 grams of bell pepper, which was already washed with the bleached and also with the water and incubated for 48 hours for the better infestation with the bacteria. This is my experimental treatment. Like control is a 0 minutes and experiment 1 for the 6 kV for the 4, 6, 8 minutes and experiment 2 for the 8 kV for the 4, 6 and 8 minutes respectively. And this is cold plasma treatment of bacteria where I have used it in UAH lab. There is a PhD student, Mr. Nageshwar, and postdoctoral researcher, Dr. Zachary White. They helped me to fix and make the arrangement of the parameters for the plasma treatment. This is how I used the plate for the bacterial sample. Bacterial growth rate measurement was done by the bioscreen analyzer, where we have seen the we can insert the microplate reader for 24 hours and we can have the data ready for the 24 hours. And preparation of Xanthomonas campestris samples for SEM analysis. For the SEM analysis, 24 well plates I have used, and at the bottom of each well I have kept black membrane and have done a lot of like buffer washed with the samples, bacterial samples, and alcohol treatment for the several washed with the chemical ethanol and HDMS for several times, and I prepared for the SEM analysis and dried the whole samples in a desiccator, stored, then sputter coated with gold for SEM analysis. This is my data analysis where I calculated mean OD differences and standard error from 3 different measurements. And I have used here Microsoft Excel calculation and analyzed the percentage changes compared to control. Results. Figure P shows the Xanthomonas campestris average growth reduction, where the treatment for the 6 kV and 8 kV for the 4, 6, 8 minutes on the growth of Xanthomonas campestris pathover vesicatoria after 23 hours. Like the control group had the highest optical density, the OD is 0.53, indicating maximum growth. Plasma treatment reduced bacterial growth with the most significant reduction observed at 8 kV for 8 minutes. The OD was 0.41. This suggests that higher voltage and the longer exposure time are more effective in suppressing bacterial growth. And the highest growth reduction 22.64 was observed with 8 kV for 8 minutes. The OD was 0.41, while the lowest 7.54% was with the 6 kV for the 4 minutes. The OD was 4.49. The results indicate that increasing both voltage and exposure time enhances the antibacterial effect of plasma treatment. This SEM pictures showed minimal bacterial suppression at lower voltage, like 6 kV for the 6 minutes and 6 kV for the 8 minutes. And control cells were they don't have changed much, and they are rod shaped only. And these control sample retained a more structured and uniform cell shape here, while treated cells began to show subtle deformation as voltage and treatment duration increased to the 8 kV for 6 minutes and 8 kV for the 8 minutes. Began significant altercations in cell structure became evident, including distortion and clustering into unusual elongated forms. This is recent study we have run again the SEM analysis where from we are got this SEM images from Dr. Bhavesh from Auburn University. Recently, this study shows the same thing with the high voltage with the 8 kV and 8 kV with the 6 minutes and 8 minutes have the pit ruptured, like cell wall ruptured bacterial cells we have seen in the SEM images. Coming to the discussions, the results from figure P show that atmospheric pressure plasma treatment reduced bacterial growth with effectiveness increasing alongside voltage and exposure time. At both 6 kV and 8 kV, longer treatment durations like 4, 6, 8 minutes led to progressively lower OD values and higher growth reduction percentages. The 8 kV treatments consistently showed greater antibacterial effects compared to 6 kV, indicating a dose-dependent response. Plasma treatment at 8 kV for 8 minutes showed the highest bacterial growth inhibition reaching up to 22.64%. These findings support the potential of atmospheric pressure plasma as an eco-friendly and effective method for microbial control, with promising applications in microbiology and related fields. Conclusion. This study demonstrated that plasma treatment effectively reduces the growth of Xanthomonas campestris pathovarvesicatoria, highlighting its potential for bacterial disease management in agriculture. Future research will explore cold plasma treatments across various microdilution concentrations to target both foodborne and human-borne pathogens, aiming to develop scalable disease control strategies. Acknowledgements. My great thanks to NSF EPSCO R2 Track 1 grants, future technologies enabled by Plasma Processing Project for providing me 50% of postdoctoral salary and funds for supplies. Thank you.